how are you? Hope things are going well. We are going to do dihybrid crosses today, which will have a lot of overlap with the monohybrid crosses. So in our for our objectives, this just goes along with number 7. So the dihybrid crosses, we'll talk about the percentages and the ratios for this one as well. And here's a little example of some mice or gerbils. I'm not sure what those are. All right. So here's the table talk question, the little discussion. You can pick either one to respond to in the worksheet below. What would you do if you're interested in 2 traits instead of just one? And then how would you figure out the gametes, things at the top? I like this picture because it takes a look at meiosis and how meiosis breaks down into the individual gametes. So there's 2 possibilities because there are 2 sets of traits. So here's one set, like if this is mom and this is dad, we've got 2 traits. So it's just letting you know that they're connected. All right, so how do you solve dihybrid cross problems? This is what a completed dihybrid cross looks like, and it has some fun little cats with long tails and short tails and coloring. So the 2 traits in this one is the coloring, whether it's brown or white, whether it has a short tail or a long tail. So determine all the possible combinations for the 2 traits. What I mean by this is how did we get these 4 gametes up here? And I also like this one with the cats because we have the sperm cells on the top and the egg cells on the bottom. So you can really tell that these would be the gametes that are created during meiosis. So how do we figure those out? We'll talk about that. After you figure out what goes on the top and the side of your Punnett square, then you have to fill in your Punnett square. This will work very similar to the monohybrid cross, but I'll give you some tips to help you keep things straight without getting all mixed up. All right. The third step would be after you have your Punnett square all filled in to take a look at either the phenotype or the genotype and figure out whether, half of them or 50% or 75% or 25% of them are a particular phenotype or genotype. As far as working questions and problems, make sure you pay attention to what the question is asking. On a test or a homework assignment, if it asks you for the genotype and you give me the phenotype, it will be incorrect. So just keep that in the back of your mind. So we are going to do a sample problem here. And if you look in the bottom left down here, we have peas, like Mendel's pea plants. And the individual little peas, they can be yellow or green, and they can be round or wrinkly. So what we are going to do is we're going to take a look at those 2 traits. So whether the pea is round or wrinkly, and then what color it is. And we are going to cross this parent, which is heterozygous for shape and heterozygous for color, with a parent who is homozygous dominant for shape and homozygous recessive for color. All right, so here's how the alleles play out. Round is dominant, wrinkled is recessive. Yellow is dominant, green is recessive. Now, I know you guys are going to be super excited about this. How many of you from math class remember the FOIL method? We're just going to use the acronym. We're not really going to do math. But the F stands for first, so this would be the first letter of each trait. Outer stands for the outer letter of each trait. Inner, and last. So I'm going to show you how to do this using parent number 1. So we've got the heterozygous parent for both traits. We're going to put that here. The first letter of each trait, if we look at this, it's a capital R and a capital Y. So I'm going to write that here, just like this. The second one is outer. So the outer letter, capital R, is on the outside and a lowercase y is on the outside. So we're going to put those here. The inner letters are right here in the middle, lowercase r, capital Y. And the last letters of each trait is the little r and the little y, which we will put here. Now, these are the gametes. It doesn't matter whether you put this on the side or the top. It's just one of the parents, so it goes on either one. It doesn't really matter. So I'm going to put them on the left side here. So I'm going to move these over and put them on the Punnett square. So that's parent 1. We need to go through the exact same process with parent 2. So on the next slide, here's parent 2. The only thing that changed was this. I know that's weird. But parent 2 is 2 capital R's and 2 lowercase r's. So let's do it again. The first letter of each trait is a capital R and a lowercase y. The outer letters, capital R, lowercase y. The inner letters right here, capital R, lowercase y. And the last letters, capital R, lowercase y. Some of you may have noticed already that this one is pretty simple. If you have, if the only option for the gametes is a capital R because it's homozygous, then you can go ahead and put a capital R for all of them. If it has homozygous for the second letter, whether it's dominant or recessive, you can go ahead and put that one. When you have a case like this, all of the gametes are going to be the same. We're not going to worry about crossing over. That would make this way too complicated. But now that you know these, you're going to put these along the top of your Punnett square. Or if you put the other ones on the top, these will go on the side. It doesn't matter. All right, so we have now determined the gametes for both parents, and we've put them on the top and the side of the Punnett square. All right, next slide. We're going to complete the square. So how do we fill in these 16 squares using the gametes here? So some things to keep in mind, some tips. When you're writing the offspring, you need to keep the letters together. So R's are first, Y's are second. And why are we doing that? The only reason is because that's how it's written for the parents. The R's were written first and the Y's were second. There's really no other reason. If it had been the other way around, we would just follow what was given to us in the problem. One thing that you always have to do, though, is the dominant letter always goes first, but you keep the letters together. So number 1 is to keep the letters together. Number 2 is to put the dominant letter first. So you'll see my example here of 2 heterozygous traits, and you'll see that that's how it's written in the parent's genome as well. Now, keep the letters in the same order as the parents. So if you have homozygous recessive R's and homozygous dominant Y's, you write the R's first. Always use the same sequence. The reason for that is because when we go to start looking at and counting up different genomes, this is technically the same thing, right? But if you just glance at it in your square, you might count them as different genotypes when actually they're the same. So keep it the same. R's always go first in this case. All right, so how do you fill it in? We're going to start with the either parent, either way. I like to start up here in the top left, and you bring that capital R all the way down. And I've color coded it to help make this a little bit easier, but you don't have to color code it. So I brought that R all the way down. Now I'm going to bring over the R's from the second parent. So there they are. Brought those over. See that? Now I'm going to do the Y's. I did the R's first because they come first in the parents' genomes. So there we have it. Now I'm going to do the R's. I like to start at the top, but I noticed that this is a recessive trait. So I'm going to put it down here, but I'm going to leave some space just in case there's a capital Y, which there happens to be over here. And then I'm going to bring these over. And there are my offspring. You'll complete the rest of the square in a similar manner. And when you're done, it looks like this. So I switch to the next slide. And after you complete it, it should look something like this. if you put the parents on opposite sides, you'll have the same number of offspring, and it'll be the same offspring. They just may not be in the same locations as mine. So, some tips about counting this up because it looks a little overwhelming, lots of R's and Y's and all that kind of stuff. So use colors and shapes to help you count. Make sure each square is only counted one time. I like doing that. If you're doing it on paper, get a couple of colored pencils or some highlighters. If you're doing it in OneNote, you can of course change the color of your pen and just circle things or put a little line through the ones that you've counted already, but make some sort of physical change on your Punnett square to let yourself know you've counted it already. So we're going to do phenotype first. So here's the phenotype. They can be round and smooth, round and wrinkly, green and smooth, or green and wrinkly. So I like to start in the top left again. So in the top left, we have a round, because remember, round is dominant. So we have a round, smooth pea, and it is yellow. All right. So I'm going to circle that. So round and yellow is what we're looking for. It doesn't matter what the second one is because those are dominant. So I'm going to look for all of the squares that have at least one capital R and one capital Y. For the wrinkly and yellow peas, it has to have two lowercase r's because it's recessive, and then it has to have at least one capital Y. For smooth and green or round and green, it has to have at least one capital R, but the color has to be homozygous recessive. For the wrinkly green pea, both traits have to be homozygous recessive. All right, so we're going to look for these combinations in our Punnett square. So let's look for all of the ones that have a capital R, at least one, and at least one capital Y. And here they are. So if you go through and look, at least one capital R and one capital Y. And we count that up. That is 8 out of 16. You need to reduce that ratio to the smallest possible, which 8 out of 16 is the same as 1 to 2. All right, then we go on and look at the two lowercase r's and capital Y, and we look through here, and we don't have that. One way you can tell is because one parent only had capital R's to donate, so there's no way we could end up with a wrinkly P. So that's 0 out of 16. That's the ratio, 0%. Now we have a round but green P. So we look for capital R with a double lowercase, like this one, or this one, which means if you count those up, it's the same as the first one, 8 out of 16. When we reduce that ratio, it's 1 to 2. And there are no homozygous recessive for both traits. So 0 out of 16. All right, so we've counted the phenotype, and that's how we do the ratio for that. Let's take a look. We'll clear the colors off of our map on the next slide, and let's take a look at the genotypes. So again, I like to start in the top left, capital R, capital Y, lowercase y. So I wrote that here. Let's do that one. There they are. Those are the only ones that occur. So the ratio is 4 to 16, which if you reduce it is 1 to 4. 1 to 4. All right. Then we have capital R, lowercase r, and two little y's. That one's down here. And I think I might have put these, made a mistake on your thing. You're going to have to fix yours too. This should be capital R, little y, because it was just like this one a moment ago. All right, so we got the first one. Make sure you fix that. 2 big R's, 2 little y's. And that is right here. And I've circled those blue, same ratio as the other one. Then I have a capital R and a lowercase y. So I put that one in my chart and then counted up all the ones that were like it. And there were 4 out of 16 again. The last one, big R, little r, and then homozygous recessive. There are 4 of those as well. So 1 to 4. And those are how you count the genotypes. So on the questions, if it's asking, I'm going to sneeze. No. If it's asking for the genotype, you're going to list the actual alleles. So you're going to see both alleles. You're going to tell me whether it's homozygous recessive, homozygous dominant, or heterozygous. If it asks for phenotype, you would literally just say round and wrinkly, or round and yellow, wrinkly and green, whatever the traits actually are. And finally, you're going to have one phenotypic ratio and one genotypic ratio. So how do you find those and how do you write them, especially for the dihybrid crosses when you have several traits? So here are the traits and the ratios that we found for the phenotype. Here's the ratios and the actual genotypes here as well. So when you write these, it can be a series of numbers. It doesn't have to be just 2. It could be more than that. And the first number is always the most frequent combination. So if you're talking about phenotypes, it's going to be whatever phenotype combination was the most frequent in the offspring. In our case, they were the same. We had a 1 to 2 and a 1 to 2 ratio. That's 50-50, right? So a ratio for 50% between these two phenotypes would be a 1 to 1 ratio. For every one of the round and yellow, you have one round and green. And that's it. That's all you have. So there's just 2 numbers in that ratio. When we look at the genotypes, however, we have 4 different genotypes that are possible. So we're going to have 4 numbers in our ratio separated by colons. The ratios in this case, just like with the phenotype, they happen to all be the same. So if they're all the same, it's going to be 1, 1, 1, 1, 1. Your homework tonight actually goes through and shows you a dihybrid cross where the phenotypes and the genotypes are not all the same percentages, and it has a little reminder how to do the ratios. But if you need help, send me an email. Bye.